like to begin my talk today by asking sort of an odd question, which is, do you think that eating currywurst can cause cancer? And it might seem kind of funny, but it's important to think about because we now know that the foods that we eat in our diet can be linked to the development of diseases like cancer. And this is largely through the modification of our DNA. So a lot of talks today have been on DNA, and I brought in a large model today just for a bit of inspiration, which you can see here. And so I'll come back to that a little bit later in my talk. And so first I want to start by taking a trip inside the cell into the area of the cell that contains all of our DNA, which is called the nucleus. And that's the black region of the screen here, the circular region. And the DNA is often termed the molecule of life. It contains all of our genetic information as a coding system that tells us if we're male or female, how tall we are, what color our hair is going to be, for example. But within the cell, there are substances in the environment that can react with DNA. And so that's represented here as these small white structures that are floating about on the screen. And so this can be components in the food, for example. And this can react with DNA, and as you can see, it can change the chemical structure of DNA. And so this is the needle in the DNA haystack. So we're trying to develop strategies to detect DNA damage. And you might be asking the question, why would we want to detect DNA damage? Why is it important? And the reason that it's important is because DNA damage is linked to carcinogenesis, or the development of cancer. And to understand this process a bit more clearly, you have to understand just very fundamentals of DNA replication. And there are certain enzymes within our cell that are responsible for making a fresh copy of DNA, so that when our cells divide, each cell has an equal copy of DNA in the parent cell and also in the daughter cell. And so these enzymes are called DNA polymerases, which is represented on the slide as this orange globular structure. And they basically work like a photocopier. So what they take is one strand of DNA as the template, and they connect together the building blocks of DNA to create a fresh new strand. And the building blocks of DNA consists of a 4-letter code, A, T, C, and G. And so basically this enzyme glues together these letters into a coding sequence that gives us information about ourselves. But the problem comes about when these enzymes encounter the site of a DNA damage, and this coding information has now changed. And often what they can do is insert the wrong letter at that position. And this creates a misspelling, if you will, or in scientific terms, a mutation. And what we've heard earlier in talks today, mutations can be advantageous. It's the basis of evolution. But they can also be bad, as they can lead to the development of cancer. And that's what we're interested in. A lot of labs that study the connection between DNA damage and cancer use strategies that look at DNA damage on a very global level. So they detect DNA damage across the genome. And we're taking a little bit of a different approach. So we're trying to detect DNA damage in very specific locations of DNA. So this is a more gene-specific approach. And we want to do this so that we can answer the question, does this DNA damage at this precise location within the DNA sequence lead to the development of cancer? And so to conceptualize this a little bit more clearly, we know that within a cell there are 3 billion base pairs of DNA, or these 3 billion pairs of letters. And so that leads to 6 billion individual letters. And if we're trying to find one DNA damage within these 6 billion letters, that would be analogous to trying to find one person in roughly the world's population. So the task at hand is a very challenging one. And one of the hurdles that we face in trying to develop these strategies is to make them sensitive enough so that we can overcome this numbers problem. And so the approach that we take in our lab to detect DNA damage is to design probe molecules that can recognize this DNA damage and give us a readout strategy so that we can detect the presence of damage at a specific location in DNA. And to illustrate this a bit more clearly, I would like to just draw your attention to this large DNA structure here. So DNA is made up of 2 strands that can come together, much like a zipper mechanism that you can see on the slide here. So it can zip and unzip together based on its sequence. And so if you can just imagine for a moment, unzipping these two strands, so do a bit of mental gymnastics. And in one strand, you have the DNA damage that you're trying to detect, so we can represent that here as a red jigsaw puzzle piece. And on the other strand, which is complementary, we have our probe-containing molecule that we can synthesize in the lab and put into DNA at a specific location. So when these two strands come together, they basically zip together, the probe molecule can recognize the DNA damage. And we have a variety of analytical techniques in the lab that we can use to detect this new unique base pair. And it's our hope that in the future, we can use these strategies to better understand the link between DNA damage and cancer, and hopefully we can answer complex questions like, does currywurst cause cancer?